Hi, we are Group 6 presenting on this study about probiotics and brain function in older adults. Aging is an inevitable process whereby your biological functions decline progressively. In fact, cognitive impairment and mood disorders are common and major health issues among older adults. Moreover, there are social issues such as high economic burden and low growth potential of an aging society. Thus, there is a need to develop efficient preventive and therapeutic strategies targeting neurodegenerative disorders to promote healthy aging. One way to do this is to enhance our gut microbiota. The composition and function of our gut microbiota changes during aging, which may affect health and age-related diseases. Research has shown that maintaining gut microbial balance is crucial for healthy aging. The gut-brain axis, which refers to bidirectional relationship between gut and brain, can link gut microbiota to age-related neurodegenerative diseases and mood disorders. Furthermore, there is growing evidence that diets can affect brain activity, making brain health preservation through dietary interventions a hot research topic. In particular, probiotics can potentially regulate brain health via the microbiota gut-brain axis. They have been shown to affect intestinal microbial dynamics and homeostasis and influence the physiology of the intestine and the brain. However, current evidence mostly comes from animal models, so scientists need to assess whether such findings are translatable to humans. Thus, this video presents the study by Kim and his team to validate the clinical properties and effects of probiotics on human gut and brain health, particularly focusing on independently living older adults who may be majorly affected by cognitive and mental disorders.
The study, which was conducted at the Korea Hospital, investigated the impact of probiotics on older adults' intestinal health, cognitive function, and mood with the hypothesis that probiotics have a positive effect. Recruitment involved individuals aged 65 years and older who underwent a screening process to gather health history, behaviors, and dietary habits information. Their physical and cognitive function status was also evaluated. Only those who agreed to random assignment and abstaining from dietary supplements and certain medications were selected. Participants were refrained from dietary supplements and probiotics during a 2-week washout phase. During the 12-week intervention period, participants maintained their usual habits, recorded their treatment intake and any unusual events. They took 4 capsules per day, 2 in the morning and 2 in the evening. In the probiotics group, each capsule contained Bifidobacterium bifidum BGN4 and Bifidobacterium longum borii in soybean oil. In contrast, the placebo group capsule contained only soybean oil. Participants were randomized and the treatment allocation remained concealed from both participants and study staff by making the treatment products indistinguishable. Blood and stool samples were collected at baseline, the 4th, 8th, and 12-week visits. Blood samples were collected in the tubes, separating serum and plasma and stored at negative 80 degrees Celsius. In contrast, stool samples were collected within 48 hours of visits using tubes with DNA stabilizing preservatives stored at room temperature and then shipped to the lab for storage at negative 80 degrees Celsius for subsequent analysis.
To evaluate the experiment results, various approaches were employed, including the examination of anthropometric measurements, assessment of intestinal health, analysis of gut microbiota, evaluation of cognitive mood and function, and the statistical analysis of data collected. For anthropometric measurements, during each visit, body weight and height were assessed using the weight scales and stadiometers. Body mass index was then computed by dividing the weight in kilograms by the square of the height in meters. To assess intestinal health, participants were asked to fill out a general health questionnaire at the 4th, 8th, and 12th week of the study. The questionnaire inquired about 10 distinct aspects of bowel health habits over the preceding 4 weeks, covering parameters such as overall bowel health, frequency of defecation, quantity of defecation, and the sensation of incomplete evacuation, and more. To assess the gut microbiota, the process involved genomic DNA extraction, amplification of the 16S rRNA gene, and subsequent sequencing. The sequencing data was then subjected to bioinformatic analysis. In order to evaluate cognitive mood and function, we used a few metrics. Cognitive function was assessed using tests designed to measure various functions, including language, memory, visual-spatial processing, attention, and execution functions. Stress levels were evaluated through a questionnaire that focused on experiences of burnout, depression, and anger during the past month, while quality of life was measured using the satisfaction with life scale. Depression levels were evaluated through a questionnaire designed for diagnosing clinical depression. The positive effect and negative effect schedule was used as a self-report instrument that helps in measuring both positive and negative effects, providing a comprehensive view of emotional well-being. Finally, statistical analysis is carried out to derive meaningful conclusions from the gathered results. Various tests will be performed to assess the normality, assumption, and homogeneity of variance, comparing variables between groups. For analyzing repeated measures, data, mixed model analysis of variance will be employed as a parametric approach, or the fragment test as a non-parametric alternative. Correlations were evaluated using the Spearman-Rank correlation analysis. Issues of multiple comparisons were corrected.
We move on to the results and interpretations. The primary findings highlights the beneficial impact of probiotic supplementation on intestinal health and gut microbial communities. In both the placebo and probiotic groups, there were no significant improvements in most bowel habits. However, as observed in figures A and B, the probiotics groups exhibited significant enhancements in gas passage frequency and abdominal distension scores compared to the placebo group with a p-value of less than 0.05. At the phylum level, there were no notable shifts in relative abundance. However, at the genus level, pro-inflammatory bacteria like Eubacterium, Alisonella, Clostridioides, and Prevotellacea demonstrated a gradual and significant decline by week 12 with a p-value of less than 0.05 as well. It's worth emphasizing that these pro-inflammatory bacteria are responsible for producing histamine, an inflammatory compound linked to autoimmune disease and chronic intestinal inflammation. Moreover, increased histamine levels in Alzheimer's patients are known to drive neuroinflammation. Therefore, the results suggest that probiotic supplementation in older adults may hold promise for addressing inflammation, which is the persistent low-level inflammation associated with aging, by fostering positive alterations in microbial composition.
Next, it was observed that by week 12, the probiotics group showed improved mental flexibility and lower stress scores, suggesting enhanced attention, executive function, and reduced mental stress. Given the positive effects of probiotic supplementations on cognitive function and stress reduction, the research team examined serum BDNF levels, which is crucial for learning, memory, and stress regulation. At week 12, the probiotics group showed a substantial BDNF increase, while the placebo group did not. To examine the link between gut bacterial changes and serum BDNF levels, the team performed a correlation analysis between the relative abundance of each bacterial genus and the BDNF levels. Eubacterium and Clostridioides show a notable negative correlation with the serum BDNF levels, particularly within the probiotics group. These findings suggest that the reduction in their abundance in the gut due to probiotic supplementation is closely associated with the increased BDNF levels, thereby enhancing brain function. The study highlights the positive impact of a 12-week probiotic intervention on serum BDNF levels, particularly among the older individuals, underscoring the potential benefits of probiotics for this population. The study findings reveal a correlation between the levels of BDNF and the relative abundance of altered gut microbes. With this relationship being specific to the probiotics group, this suggests a potential influence of probiotics on the interaction between the gut microbiome and the host BDNF, which may contribute to enhanced brain function. Overall, the evidence suggests a relationship between changes in microbial community and alterations in cognitive and mental stores, implying that the gut microbiota could play a role in cognitive and mental well-being.
As to all studies, there are always limitations and improvements on future works. Here are the limitations and potentially the future studies that can be done. First, direct evidence of improvement in peripheral and cerebral inflammation by probiotic consumption is lacking. This is a crucial interface linking the gut and brain axis. Further studies needed to elucidate the role of probiotic supplementation by finding biomarkers to link the gut-brain axis. Second, some cognitive functions in the neuropsychological assessment battery task had no significant changes. The researchers speculate that this was because the study duration was not long enough to monitor such improvements. Therefore, further studies with longer period of intervention are required to capture the longer-term impacts of probiotics on brain function. Third, although the battery tasks were done by a panel of professionals, the psychological assessments of mood status were based on the participant self-reporting. Hence, possible recall bias may exist. Fourth, the small sample size of 63 adults. This can limit the generalizability of the findings and the ability to detect smaller or less common effects. Future studies can be conducted with a larger sample size. Fifth, there can be variations in the result with different probiotic strains, dosages, and formulations. The study may not be able to determine the optimal strain, dosage, or treatment duration required for meaningful effects, as it only uses Bifidobacterium bifidum BGN4 and Bifidobacterium longibori. Lastly, this is the first ever study done to examine the effects of probiotic supplementation on brain functions in community-dwelling older adults. Hence, there is a lack of reference or existing knowledge from previously done or similar studies. To conclude, the 12-week study on older adults' consumption of probiotics was found to have a noteworthy correlation in the improved cognitive and cerebral functions of the brain, potentially as a result of the reduction in pro-inflammatory bacteria, which in turn increases the BDNF levels. Hence, probiotic use may enhance the gut microbiome's interaction with BDNF, leading to an improved brain function. Hope you enjoyed our video. Thank you.